So we did DNA replication, and DNA is found in the chromosomes of all organisms and is the genetic material for all organisms. And so before the DNA, so when the DNA replicates, first the helicase comes and separates the two strands of DNA by breaking the hydrogen bonds into replication fork, which is this like V-shaped region, and then polymerase III comes. The single-stranded binding proteins attached to the strands of DNA so that they don't want to come back together. And then polymerase III comes, and since this is going from 5' to 3', it's moving that way, and it's laying down the corresponding nucleotides for the leading strand.
On the lagging strand down here, it starts with the RNA primase, which follows the helicase this way, and it lays down RNA primers, which are these represented by these. And the RNA primers signal the DNA polymerase III to come over and to start laying down the correct bases. So since it's 5' to 3', it starts here, and it lays it down that way. And then when it's finished, when it reaches another one of the RNA primers, it comes back this way and begins replicating this way.
And I just had general notes about DNA itself. So up on this section right over here, if you go past the replication fork, DNA is actually composed of these things called nucleotides, which is made up of a sugar, a phosphate group, and hydrogen bases. So basically what you have, the nitrogenic bases are composed up of hydrogen atoms, nitrogen atoms obviously, and carbon atoms, and what you have is adenine, thymine, guanine, and cytosine. And so the adenine and the thymine will always go together as a base pair because you're always going to have a pyrimidine and a purine come together. A purine has a double nitrogen ring around it, and a pyrimidine only has a single ring. So that's what you have over here in this section.
And to finish up the lagging strand, to replace the RNA primers, the DNA polymerase I comes, and I don't know if I want to take this out, but it replaces it here. Okay. It goes this way and replaces it like that, and then it would come back over here once this is finished.
And then the ligase attaches all of the Okazaki fragments together to give the clue.
Yes.
For the lagging strand.